Hi, good evening all. A warm welcome to the today's webinar on optical tweezers for biological research from single molecules to live cells. And I hope everybody is doing good and having a sound health conditions during these pandemic situations. Core Vision welcomes you for today's webinar, and I would like to introduce about today's speaker, Dr. Yogesh Jairam, and he serves as a key accounts manager for Bruker Nano Surfaces in India, and he is responsible for application development and technical support of surface metrology and tribology instruments. For his academic background, he was pursued his PhD in solid state physics and material science, and he pursued his PhD from KU Leuven, Belgium, working on linear and nonlinear effects in metal nanostructures. Prior to joining Bruker, he has worked as a postdoctoral researcher in University of Toronto. He has published over 15 peer-reviewed international journal papers. He also presented at many international conferences as a speaker. And before inviting Dr. Yogesh Jairam to present his talk, I would like to grab the attention of viewers. If you have any questions, please type your questions in the Q&A section, right side of your screen. You can type in your questions and we will get back to you with answers. If the time doesn't permit and your questions were not answered, please don't worry. We will get back through you email separately for you. And with this, I would like to welcome Dr. Yogesh Jairam to take over the session. Dr. Yogesh.
Hi. Yeah, good afternoon. Thanks, Raghav, for the introduction. Good afternoon, everyone. Thanks for making time and joining us today. I would like to talk about this new topic called optical tweezers. So many of you have been familiar with it in terms of science, but we'll be more talking about applications of it, more towards how this product is put together by Bruker and how it's being used by some of our customers for various research in biology, right? So from single molecules and live cells, etc. Okay. Okay. Let me see. All right. Okay. So optical tweezer, a Nobel Prize winning invention, right? So some of you might be familiar that Arthur Ashkin got a Nobel Prize for optical tweezer. He shared that with Gerard Moreau and Donna Strickland for their work in the lasers as well. So the concept of optical tweezer is so that you have, let's say, a transparent bead, and then when you shine a laser through it, and if you have like a focus point, at the focus point, you're going to have some force gradients, and this is going to make the bead behave like it's attached to a spring, right? And the reasons that when you move the laser, so the force is going to be proportional to the displacement, and it's going to give you very similar force behavior to Hooke's law, right? And that's what we are leveraged to make this tool, and this has some fundamentally great applications as well in terms of fundamental research. So you can now trap transparent molecules or transparent beads and other things, and you can move them around in 3D, and you can do various experiments with them, right? So when it comes to biological concepts, so we can look at the 4 scales of different molecules. I mean, if you look at single molecule spectroscopy, I'm talking like a protein or a single DNA strand. So we are talking the force ranging from anywhere from 1 piconewton, so maybe to 2 piconewton. When you talk about cellular mechanics, how stiff the cell is, what's the adhesion strengths, etc. So we are talking a little bit larger forces, so maybe about 20 to 13 piconewtons, all the way to maybe 10 nanonewtons or above. So I mean, even though we are talking about these as large forces, these are fundamentally very, very small forces, right? I mean, 1 piconewton is 10 to the power minus 12. So it's a very, very small force. And to be able to control an object at that force levels is a technological challenge. And this has been addressed predominantly by two techniques, right? So the atomic force microscopy is a good technique, so where you have like a cantilever and you use the cantilever deflection dynamics to understand the force and how the behavior on a single molecule or a cell. And it has been used extensively to study this biological world. The force here, yeah, it can be from maybe few piconewtons to maybe 10 nanonewtons or even larger forces is possible with AFM, right? But if you look at the optical tweezers, then of course your force resolution is much, much smaller. So now you can get like 0.1 piconewton to 100 piconewton. So this makes the optical tweezer a unique tool in that the resolution of this tool is much smaller, right? I mean, at least a 10 times smaller than the dynamics that you are interested in studying. For example, the single molecule force spectroscopy. And that's where it becomes its own, right? When you are studying very, very small force interactions, the optical tweezer has the resolution in terms of force to help you study that without concern for noise or artifacts. They are introduced by the instrumentation technique itself. And that's why people use that on top of the AFMs as well. AFM is still widely used for single molecules and cell studies. The optical tweezers add a dimension to that. Okay. So an optical tweezer looks like this. Well, this is a commercial setup. So maybe some of you have seen a home build setup with a lot of the open ports and other things. So these are nicely concealed. So all your steering mechanisms for your traps and your lasers are inside this. And this is your conventional inverted optical microscope. And then you have your samples here. And then you have the detector set here, right? And everything is controlled through the computers. Okay. So what's inside this white box, right? What's inside this white box? Let's have a look at that. Okay. Sorry, something in my screen. Okay, let's put that there. Okay. If you look at that, there is a laser, so you can have different power lasers. So then you have some way to control the power, right? You have the shutters, and then you split the beam into two. So one can become the trap 1, trap 2. So you can have two independent traps in our system. One can be static or moving, the other moving. Yeah, so you can choose if you want both the traps to be able to move in 3 dimensions. Or you want one of them fixed permanently. And then you can, this light is fed through a proprietary port, and then it goes to the sample, and then you can do the studies on your samples, right? And you have a very sophisticated detection package, which helps you detect the position and the force in 3 dimensions, right? So you're moving your trap in 3 dimensions, you will generate the force in 3 dimensions. You can study the positioning of the traps and your forces in 3 dimensions. So it gives you that. And you can see here the way this has been done with our dedicated port here. It doesn't affect any of your fundamental ports of your IOM, which may be here or there, where you can connect your laser, power sources. And it also doesn't, it's above your filter wheel, right? So it also benefits from that. Okay. So the typical laser we use in all our cases is a 1064 nanometer. It's particularly chosen, it has a low absorption in biological sample. That's one of the reasons we use that. But of course, one can engineer for any laser wavelength, the optical trapping. Okay. So how do we move the traps? How do we control the trap positioning? There are two major techniques available. One is a piezo mirror. So this is, yeah, of course, many of you are familiar with piezo. So you apply electric bias, it moves, and you can get some nanometer level accuracy in terms of positions, right? And can have also large range. Then if you want to have faster dynamics, if you want to move the trap very fast, the piezo generally doesn't have the bandwidth to it. So if you want to have ultra fast movement, then people generally choose acoustic optic deflectors or AODs with over 25 kilohertz position updates, right? So it moves the traps very fast. It's very especially useful if you're doing something when you're studying something which has a faster dynamics. Okay. So if you look at the optical tweezer applications, which we call the nanotracker, so you can largely divide them into three areas, right? So single molecule studies, living cells, and materials research. So of this, the biology is a larger section. So many publications are there, many people use it. Right? So if you look DNA mechanics, DNA protein interactions, right? Or if you are looking at a motor protein, you're looking at how the motor protein is moving and what's the kind of force it generates, right? So to understand this behavior, also for protein folding, unfolding studies, RNA hairpins, etc. Right? When it comes to living cells, there's plenty of things to study. So you can talk about virus-cell interaction, a bacteria-cell interaction, or a bacteria-bacteria interactions, right? That's studying interactions. Then you can study the cells itself. What are the mechanics, and how the membranes are moving, or how the cytoskeleton adhesion, or how it's stuck to the membranes. And also study immunology, right? So if you can study virus-cell interaction, you can also study how the immunology, cell immunology is there. And also you can do in-cell manipulations and measurements, right? Then in materials research, people study mostly rheology, right? So if you want to study viscous material, so, like biological materials are viscous, so if you're studying viscous material, so you can study the viscosity and then how the property changes in water or different, solvents. You can do this kind of analysis, right? Okay. So let's start with the single molecule studies. If you think about that, so if you have like a DNA, let's take it for example, and then you want to attach some beads to it, right? If you do it in a conventional way, so you do it in your lab, there's a large chance of it getting agglomerated. The beads, maybe you get two, maybe you get many. It's all kind of like a mismatch. So then you have to sort through it under your microscope to find statistically, well, some DNA has magically two beads that's out there, both ends. Then you pick that and do the studies. You can do that. So the nicer way to do it is we have this kind of like a Y kind of channels. So on one side you flow all the DNA, on the other side you flow the beads, right? Which are already coated with the molecule so they can bind to the DNA. So now these are flowing and they're not intermixing with each other, right? Because of the way the microfluidic device is constructed. Then you can pick up, let's say, two beads and then bring it together and then, attach it to a DNA on there. So you can very clearly do that without any problem of agglomeration and other things, right? So you have this combination. And this is, of course, fully temperature controlled and flow controlled. So we're able to do that. Okay. So once you do such an experiment, then you have this two beads and you, let's say, have a DNA in between. I'll show you like a small video. So you can see here, well, the DNA, this is a cartoon, yeah? So you're moving one bead is fixed, right? So you're pulling the DNA up and down, right? So you can see that you're moving the DNA up and down. And then from there, you are here. In this case, you're moving from the entropic regime to the enthalpic regime. And then if you stretch it long enough, then you go into the overstretching transition, right? So you can see here that you cannot see the DNA because it's unmarked and it doesn't have any fluorescent signal. But you can see we are pulling a DNA by looking at the bead one, which is supposed to be static. But then when you get into the overstretching regime, maybe you can watch it again, and get into the overstretching regime, it will move away from the center position, yeah? So this shows you that there is a molecule that we are pulling, and of course the force data also tells you that, right? Okay, so then you can also do other interesting studies here. So in this case, you can clamp it. It's called force clamping experiments. So what you do is you pull a molecule or a DNA or whatever, and then you hold it at a particular force, right? So when you hold it at a particular force, the way it interacts with other molecules changes, right? So if you say, for example, I have a protein in a no force condition, nothing happens, then it stays same. Then when you clamp it at say at 20 piconewtons, the way it interacts with other molecules or cells is going to change, right? And this is happening in our bodies as well. It's one of the mechanism. Force or mechanical stimuli is one way that certain reactions are turned on or off, right? So you put your molecule in stress or relax, you will trigger certain kinds of reactions in your body and maybe some immunoresponses as well. So that's one way people use to study, right? And because you have like super controls with force, like sub-piconewton force control, you can comfortably do that without the force getting drifted away. Yeah. Okay. So you can see here the force control is less than 0.5 piconewtons, right? So excellent force control that we can get. So other thing that we can study, like I mentioned earlier, is on the motor proteins, right? So the motor proteins generally moves on the microtubules. So you see, you can attach, let's say, a bead on top of your motor protein, and then you hold it, right? But you don't apply too much force. But this motor protein now will move on top of your microtubules, right? So then you are just tracking your bead, and then you can see what's the speed of your motor protein movement and what kind of forces it's having to complete the movement. Yeah. The general problem, if you use a stationary bead in this case, is you are limited to studying only a certain time durations. Yeah. So that means you cannot when the motor protein moves far enough, it completely comes out of your trap. Okay, so when all right, so if you have the trap that's also moving along with the motor protein, then you can study these interactions for a longer time, right? So if you have a trap that has the flexibility to move and it's fast enough to track that, then you can do that. And that's where, all the piezo mirrors and the AODs that we talked about come into play. So I have some questions here. I will take them when I can. So there are some questions, for example, how do you attach beads to the DNA as both are moving? How can one measure the stretching force in this case? So you attach the bead, you trap the beads first. The beads are moving, then you trap it. Once you trap the beads are not moving, right? And beads also have molecules on top of them, which attract themselves to the DNA. So you trap the beads, you move them to the DNA side, like in the fluidic chamber that we showed, and then you wait for a DNA to come. You control the flow rate so that it's something reactable too, right? It's not like ultra fast. And then you hold it there, and it will automatically attach to the DNA. Then you can hold this. Now you have DNA attached to one bead. Then you trap another bead and bring it there, and you can attach it to the other side of the DNA as well. And that's how you do it. And once you've done that, you can hold the DNA and you can flush your channels, and then you isolate the DNA in a static condition, no flow conditions. Then you can do your stretching experiments, and that's how you do it. And there are many creative ways to play this. Yeah, so your limitations is only our imaginations, right? Okay. So when it comes to cell studies, so one of the interesting things that we like to study is the membrane tether pulling, it's called. So what people do is we attach a cell to a bead, right? The bead is now attached to the cell, and then you pull the bead, right, away from the cell. So this pulling is what you pull is the cell membrane, is to study, the mechanical properties of the lipid membranes of the cells, right? That's the main idea. So you can see here on the microscope, you're pulling the bead away from the cell. I do not know whether you can see, well, there is some contrast here on the membrane tether, right? And the picture above shows a better contrast of that. So you have the beads here, you have the tether there. What happens, in the cell is when these membranes are attached to the cytoskeletons, right? So initially, when you pull, you have to detach the membrane from the cytoskeleton, but then you can pull it. So this can be about 10 nanometers or 100 nanometers, depending upon it, and you start pulling it, then you can study that. So if you, let's say for here, you can see if you attach only to the lipid membrane, there is no cytoskeleton involved, you have this kind of behavior. So this is the amount of force and discontinuous. But whereas if it has the cytoskeleton, you will have to disengage from the cytoskeleton, then you are starting pulling the membrane tether, right? And that's what people do. And once you do that, you can study what's the effective membrane viscosity is, what's the tension behaviors are, and there are many publications on this and how to study what kind of analysis that we can perform there. Typically, in real life, you end up attaching more than one tether or one membrane point to the beads. And this is what you will see, like this step. So you have multiple membranes attaching, and then you have some disconnecting, then you have a drop in force, then you have another one drop in force, and the second membrane connects, and then you have another drop in force, and the final one disconnects, yeah. So this is common. So it's people study that as well. Yeah. So then, how to study the cell itself? Because mechanical properties of the cell, in this case, the red blood cell is integral part of the cell function, right? So let's say healthy red blood cell will have a completely different mechanical properties to an infected one, like malaria or other diseases. And also, if you're looking at the bacterial cells modulus or the elastic modulus changes depending upon whether the cell is, undergoing cell division, or it's and which phase of the life the cell is. It's all dependent, right? Also, now there's a lot of work that shows, like how the cell development, like even stem cells, they are even very sensitive to the surroundings mechanical properties, right? So if you put a stem cell on a really hard substrate, the cell will grow into like a bone cell. But if you put on a very, very soft substrate, they will grow into like neuronal cells. So there's a lot of interest in understanding the mechanical properties of cell, particularly the modulus of it. So in this case, you have a unique thing, right? So you have a beads, which is very spherical, and then you can indent the cell, and then you can study the force dynamics, right? So when you are indenting, the cell opposes that, and then you have the center position of your trap changes from the bead center position. And from this, you can calculate what kind of forces you have. And you also have the ability to control the indentation depths, right? Measure the indentation depths. And then you get this kind of plots from which you can calculate with simple formulas what the Young's modulus is of your material. But you know that the cells are not only elastic, they are viscoelastic. So it also, the modulus or the mechanic property, it depends on the loading rate, right? And that's very important to study as well. To do such experiments, what researchers have done is they attach two beads. So let's say red blood cell in this case, then you attach two beads, one on the opposite sides of each other. Bead one is kept stationary, and the bead two is oscillating, right, at a fixed frequency. You move the trap, the bead responds to it. So that's what you see here in this graph. So even though the bead one is supposed to be stationary, so it still responds a little bit to your applied stimuli, right? Because the other bead is pulling it. Then you oscillate the trap, so you are moving the trap. That's the red line, and that the bead two's response to the movement of the trap is the solid black lines, right? And then response of the bead 2 movement is responded by the bead 1, right? So here you have a small video that shows, you have one trap that's fixed, more or less fixed. The other trap is moving, and you can see its different frequency, first initially slowly, then at a higher speed, right? Let's play that again. So initially it's moving a bit slowly, then you see that it moves at a higher speed, right? Okay, so you can see here, so when you're oscillating at a lower excitation frequencies, your bead 1 and bead 2 is moving, in phase. But where high frequencies, they are out of phase, yeah? And this is typical for this kind of materials you study, and you can apply different models to understand that. We have some standard models that are there that you can use to study, especially when it comes to, like, say, for example, single molecule, you have like worm-like chain functions, and you have different models for that, and you can use the models, you can get the force data, and then you can use it to study that. Yeah, and we measure, yeah, I mean, everything is calibrated, so you measure the force in newtons, right? And you measure the position in nanometers or micrometers or whatever, then everything is in SI units, and then you can directly fit the model. The software will do it for you, and then you can play with it. And in many cases, as a researcher, you might need to use your own models, because this is still research area, right? It's not settled science. Then you can get the data, and you can fit different models of your own to study that as well. So in biology, it's very common that you need a fluorescence signal to highlight a region, or to find the region of your interest, right? Otherwise, there's not much contrast in biological material, even with DIC in some cases, right? The unique setup of our system gives you a full range of freedom. So you have the source, and you can see our laser, or where we enter from with our laser, is above your fluorescence filter carousel. So you can move the filter without actually moving, affecting the trap. So that's what you see in the video. This just to show you. So you have a trap that's moving in circle. Yeah, you have a fluorescence molecule that's trapped, and then you can see we are moving in and out the filter, but we are in no way affecting the trap. Right, so you can turn on or off or move your filter carousel without affecting the track positioning at all. And this gives a lot of freedom, right? Gives you a lot of freedom to do that. So yeah, like I said, our trap, the way we enter the light is much higher than your filter cubes, turrets, and your carousel. So it doesn't get affected if you change anything below that, no problem. Right? Okay. You also can integrate it with the total internal reflections, where the laser comes at a very high angle, and it has a very low penetration depth in your sample, so it's totally reflected. So only the molecules that's very close to the substrate will have a fluorescence signal. So this lets you, capture fluorescence data without much background noise, right? And that's why people use it, and you can use it along with our trapping optical tweezers as well, right? So it's also doable. When you are integrating, let's say, a fluorescence, but you're integrating with a confocal, yeah, now your trap can move XYZ, and your sample can also do XYZ movement, right? In a confocal laser scanning microscopy, you have a laser that's scanning the sample, like rastering it, and you are moving in Z, and you have a pinhole. So that means wherever you're absolutely focused, only that region is captured, right? And you have like multiple Z levels. So here you can see, well, so yeah, you can see in this video, so you're moving the molecules in X, Y, and Z. Yeah, that's why the sample goes out of focus and comes in focus. And then the trap moment is independent of your sample, right? And you can move your sample independent of the trap. So you fix your trap and you can move your sample. So this is the trap molecule, also the trap bead. The sample is on the background. There are other beads also like this one. These are not trapped, yeah. So these are moving whatever they want to do. So this bead is trapped. And you can see you can move X, Y, Z, the sample, and the trap in X, Y, Z. One is not getting affected by the other. It gives you a great amount of freedom to play and position the bead exactly in the 3D universe, right, where you want it to be. Okay. When you do the force measurements, you can, like we discussed, move the bead, right? So you can move the bead. Let's see where it's coming. Yeah, you can move the bead and indent the cell, right? This is okay. So you get how the force behavior is over time, and you have the distance as well. Or you can move the cell, right? You're moving the sample and keeping the trap at a fixed position, and the results are the same. Yeah. Okay. So we talked about the optical tweezers as well. You can integrate it also with AFM, right? Like right at the beginning when we talked about the force region that we're interested in, we talked about how AFM also has a role to play, and now we can combine your optical tweezers with your AFM head on top of a microscope, which is all the biologists need, and you can perform more complex experiments, right? So for example, your AFM cantilever can provide a stimuli, right? So you can have a force stimuli, and then you can bring a molecule close and nearby to see how that behavior is. Or, for example, you can pull your a cell or a molecule through your tweezers, and you can use the AFM to image how the topography is, right? So now you can do topography, your mechanical properties, you can do tensile, you can do compressive forces. You have 3 different force directions, no problem, right? And you can get high quality data in a much more complex interactive way. And you also have your optical data to overlay on top of that, right? So here is, for example, a study that we did. So you have an AFM, which has a conventional T cell attached to that, AFM cantilever. You have the dendritic cell, and you have a regulatory T cell, right? So what we are looking here, we are studying is what's the detachment work that we need to do from the conventional T cell on top of your dendritic cell, and what's the role of the regulatory T cell in this mechanism. So you can see before you have the regulatory T cell in contact with your dendritic cells, you have this amount of work needs to be done to detach. So once the regulatory cell is lower, then the force suddenly drops, right? Almost halves to detach. And then when you remove the regulatory cell, it recovers, but it's still not the same as before, right? So this is the kind of experiments people study. So, how the T cells, dendritic cells, how the various cells in your body interact, what kind of relationship they have with one another, what kind of regulatory mechanism they have with one another. The biologists know a lot about them. They have done many experiments to understand it. And now you can test many hypotheses or understand these concepts in more or deeper way, right? That's the idea for it. Yeah. So just to summarize, so nanotrack is a high resolution force measurement platform with versatile applications. So extremely low noise, right? So that's the main driver. So you have very, very low noise when you are studying force spectroscopy, especially for single molecules, sub-piconewtons force controls. You have excellent fluorescence integration, confocal integration. So you have advanced environmental control. I didn't show anything. So here you have a slide here. So you have this kind of like petri dish holders with heaters inbuilt, and also, for example, CO2 and humidity controllers attached. So you can control the living environments of your cell to a great degree, so you can do very, very long life experiments, right, where the cells are happy, healthy, and you are performing your experiments over a long period of time. As a very flexible software, natively supports many wide range of applications, and also can give flexibility for users to study or extract the data to have their own analysis. Yeah, so that's it from my side. So it's a short presentation. So if you have any questions, we can talk, and then maybe we can go back to some of the slides. So you can drop your message in the chat box, and then I can read them and I can try to answer them as they come.
Hi, Yogesh. I have seen a question in the question and answer session.
Oh, is that a separate from chat box? Oh, I'm
Yes.
sorry about that. Okay, yeah.
So, how it was written by Shesha Vempati. How do you steer the laser beam? Is it by some electromagnetic lens or galvanic lens?
Yeah, we have a piezo and AOD controllers to do that. So you can have a piezo or AOD ones. Let's see if I can find that. So this one. So this is the steer. So your mirrors are fixed to either the piezo mirrors or your AOD controllers, and this is how you control it. So the AODs or piezo is depending upon your speed requirements. If you want a faster dynamic responses, AOD is the choice. And you're not. I mean, like we are talking about 25 kilohertz, right? So maybe if you're a few kilohertz, then maybe piezo mirror is fast enough for that. So that's a control mechanism for the mirrors. Yeah. So in this cross section, let's see. Yeah. So let's see if I can bring my pointers. Yeah. So this is where your controller is with the mirror. So this is AOD or a piezo. So this is for trap 1 and the trap 2 is over there. So you can have different ones there. So this can be a piezo, this can be AOD, or it can be even fixed. So then you have a fixed trap. So yeah.
Yeah. Thanks, Dr. Yogesh. I hope this will answer your question. And we will wait for a couple of more minutes to see any more questions come to us. If not, then we can close it with a vote of thanks to Dr. Yogesh. Give us a couple of minutes, Dr. Yogesh, so that we will wait for some more questions.
Yeah, sure. Take your time.
Yeah, thank you. Yeah. So as there are no further questions, uh, I would like to thank. We
Raghav,
would like to. Yeah.
yeah, Mr. Raghav, we got one another question from Mr. Brijesh Kumar Singh. He's
Okay.
asking single QPD is sufficient to measure the position, or do we need two QPD for two beam path? I will.
Yeah, so yeah, sure. I mean, yeah,
Yeah.
you need the appropriate amount of QPDs to track your beams in different directions. So if you have two traps that's moving, you don't. I don't think so. One QPD is sufficient to do that. But yeah, the detectors that we have takes care of it. So it has enough QPDs and other things to track two independent traps that's moving in 3D, both in position and force, right? So yeah, being a commercial setup, so this is all inbuilt in the engineering process. Okay.
Okay, guys. Thank you for your query, and thank you for your session.
Yeah. Sardi, is there any more questions?
No, Raghav, there is no any other questions. Only this is the last one.
Okay, so as there are no questions from the audience, we would like to thank Dr. Yogesh for his wonderful presentation with a lot of information which will help the researchers to understand the new technology, and also they can develop or they can contribute some more towards the.
Yogesh, sorry to interrupt you. I got it. Another question from Brijesh Kumar Singh. He asked
We can take it over offline, Sarvati. Let's just,
okay.
we can send a reply later.
Yeah.
No need to.
Okay.
Okay.
Okay. Okay.
So it's a wonderful discussion for optical tweezers, and we also thank each and individual participant for spending their time and learning these enthusiastic optical tweezers technique. So with this, we would like to conclude and thank you everyone for making the this webinar great success. Thank you one and all.